Hello everyone. It's a great pleasure to have you here for our second webinar in the UNIgreen's Biochemistry and Biotechnology webinar series. Today we will dive into the subject of biotechnological advances in monitoring pollution in the environment. And we have two speakers today that will help us to discover this subject. So before we go into science, just a few forwards. So I'm honored to be your moderator today. I'm an associate professor at SupBiotech, and I'm co-coordinator of research and innovation at UNIgreen University. And if you want to contact me, if you have any question, you can find here my email address. A few disclaimers. This event will be recorded and uploaded in our UNIgreen YouTube channel. And if you are a student and you need a certificate for this webinar, just contact us at this address here, unigreen.communication@gmail.com. And at any time, feel free to use the chat to ask your question about the subjects. About UNIgreen, you might not all be aware about what is UNIgreen. So UNIgreen is an alliance between eight higher education institutions across Europe. This is a program supported by the Erasmus Plus program. And our university is dedicated to the education and research in the field of sustainable agriculture, green biotechnology, and environmental science. And of course, we are targeted to deliver the high standards of education, foster research, and provide opportunity for both researchers and students and staff. Just a few words about our research activity in relation with education. So we have within UNIgreen 8 axes of research that are mentioned here. They are divided in what we are calling Joint Research Center, and today we are in Joint Research Center 1, dedicated to biochemistry and biotechnology. And so we have other that you can find here. And if you need any information about our activity, you have the email address or our website over there. And again, my email address if you want to contact me as well. So UNIgreen Biochemistry and Biotechnology webinars. It's a program. So we started in April this year with the first webinar about valorization of natural products and waste from agriculture. You can find this webinar on YouTube channel, our YouTube channel. And today, so we are going to speak about monitoring pollution in the environment. And you can see here the conference to come in September, October, November, and December. So stay in touch, contact us if you would like to see other kind of subjects. And again, here is my email address. So in today's agenda, we will have two presentations, 40 minutes more or less of the presentation, and we will have time for the question, and we can proceed then for discussion and some conclusion and closing. Our speakers today, so we are really honored to welcome Frank Yates, who is Director of Research at Biotech. He is working on neurodegenerative diseases using iPS cells and cerebral organoids, or what we call also membranes. And he will give a talk about these specific models that are very useful. And we will next have Torben Osterlund, who is the Chief Scientific Officer at Pepper. So Pepper is a non-profit association. It's a very interesting structure that is developing new assays to evaluate pollution in the environment, and they are specialized in detection of endocrine disruptors. So Torben is not here yet because he works in trouble with Metro in Paris, but he will join us So I will give the stage to Frank Yates for his talk. Just give me the time to switch the presentation.
Thank you, Patrick. So hello to you in the room, students coming to the 2025 Summer School organized at Biotech on the theme of One Health. I'm very honored to do this presentation today to show a little what we do in our research laboratory and also to discuss a very interesting subject which is actually a lot talked about in the current news. So today we'll be talking about stem cells, as you will see, and organoids, and in specific domain environmental research. You will see there are a lot of promises for this type of research, but I hope you will remember that there are also a lot of difficulties in this type of research, but it wouldn't be research if there weren't any difficulties. So, first to present myself, I'm a professor at SupBiotech, and I head a research lab in collaboration with the CERA. CERA is a French institution, not very far away from here, and we've been working for nearly 10 years now on stem cells and organoids to model neurodegenerative diseases. I'm also secretary for the French Society of Stem Cell Research, which organizes a conference every year, and you are welcome to come. Our next meeting will be in November 2025 if you're still interested in stem cells after my presentation. So, as you have guessed, I'm a big fan of stem cells, and there are some things that are very important to know when you work on stem cells. Stem cells have two properties. The first property is self-renewal. Self-renewal means that stem cells have this incredible property of being able to replicate themselves. One stem cell can give one or two new stem cells. This is how stem cells renew during life. This is how, for example, the blood stem cells, the hematopoietic stem cells that are in the bone marrow of the body, replenish themselves once they put their job. Secondly, stem cells can differentiate. They first differentiate into a progenitor cell, and then they can differentiate into terminally differentiated cells. This is what I represented here in these colored cells. Now, a property of somatic stem cells, such as the stem cells that I was talking about, that you can find in the blood, is that these cells are only differentiated into a single ligand. A blood stem cell can only give blood. A blood stem cell cannot give neurons, muscle, etc. Okay? A liver stem cell can only give cells of the liver. Okay? We say that somatic stem cells, the stem cells in our body, are restricted to the livers of which they are, which is already right. You can find stem cells just about everywhere in the body. I'm sure there are many sources of stem cells in the body that have not yet been found. The most known stem cell, as you have guessed, is the hematopoietic stem cell, the stem cell that you find in the bone marrow. We also know about a lot about the stem cells inside the liver, inside the gut, inside the skin, etc. Okay? These are called somatic stem cells, somatic means from the body. We also call them sometimes adult stem cells because you find them in the adult environments. Now, I'm a big fan of stem cells, and I have to talk to you about the Taylor Swift of stem cells, the the big star of stem cells. Sorry if this reference is a little dated. I'm over 50 now, so I don't know who's the latest star. But my star in the lab is pluripotent stem cells. Now, pluripotent stem cells, they can still self-renew. This is one important property of stem cells, but they can also differentiate in all lineages. So they are not restricted to one lineage. So a pluripotent stem cell can differentiate into all the cells of the body. Cells from the endoderm, for example, the liver, the gut, the pancreas. They can also differentiate into mesoderm. Mesoderm are the bone, the muscle, the blood, and they can also differentiate into ectoderm. Ectoderm is the neurons, the skin. Okay, maybe you remember these terms from your embryology class. Okay, now there's three germ layers that appear very early during embryogenesis. So a single stem cell that I can culture in a dish can give rise to all these lineages, and they can also self-renew. So I hope you understand how exciting these cells are. Where can we find these stem cells? As I said, there are no pluripotent stem cells in the adult. To find these stem cells, we have to start, at least that was what was first described in 1998, from an embryo. What James Thompson did in 1998 was that he took a human embryo, a 5-day post-fertilization human embryo. He took the inner cell mass of the embryo and cultured it in vitro, and he observed that these cells inside the human blastocyst could derive into cells that could be cultured in the dish that we call human pluripotent stem cells. He called them embryonic stem cells, okay? Human embryonic stem cells, HDS cells. This was a big news at the time. In 1998, when he discovered this, scientists all over the world were very excited. At last, we could get any cell in a dish, muscle, pancreas, whatever, just starting from a few cells of the human blastocyst, and because these cells can self-renew, we could get a really infinite number of them. Even more exciting, about 10 years later, Yamanaka came and invented an extraordinary invention he called reprogramming. He got the Nobel Prize for that in 2012. Reprogramming consists in taking the terminally differentiated cell from any of you, I can take the skin cell from any of you, and reprogram them expressing 4 factors that we call the Yamanaka factors in the lab. And these cells become pluripotent. Okay? So a cell that is terminally differentiated can become pluripotent and can then become any cell in the body. Okay? Pluripotent stem cells, they all look the same. There may be some slight differences between human embryonic stem cells and induced pluripotent stem cells. Induced pluripotent stem cells is the name of the cells that are reprogrammed from the Yamanaka factors. Okay? But these cells are pluripotent. Under the microscope, they all look the same. They grow in colonies, such as this. In this colony, we have 1,000 cells all living together. Stem cells, like pluripotent stem cells, really like to live together. If you separate them one from the other, they start to die, in as much they are reunion. And they can be cultured in the dish for, if you take care of them, for many months, if not years. These cells can differentiate into different germ lineages, neurons, blood, pancreas, etc., as I said. Of course, differentiation procedure is the key of this technology. We always represent it with an arrow like this. It seems very simple. It is a bit more difficult. We have to use media, we have to use proteins, inhibitors to convince the pluripotent stem cells to become differentiated cells. And now I want to address a particular important fact, is that there are two types of cell culture. I'm sure you've all done cell culture in labs, whether in internships, in practicals. Indeed, you students at the summer school, you are currently doing two-dimensional culture of cancer cells at the moment in the labs. What we call two-dimensional culture is culture in a dish. Okay? Here's a dish. You can imagine the cells growing at the bottom of this dish. They are attached. And of course, there's medium. There's a medium in order to bring to the cells the growth factors, the sugars, and all the other elements that need to grow. Now, this is two-dimensional culture. Okay? A thin layer of cultures. And that's already great. You can have, at the bottom of this dish, you can have muscle, you can have neurons. But thanks to pluripotent stem cells came a new way of culturing cells. I'm saying new, people have been growing cells in 3 dimensions for quite a while now. But now it brought it to everyone. Everyone could grow cells in 3 dimensions. And you will agree that 3 dimensions represents better a tissue, an organ. Okay? Our brain is 3-dimensional, it's not a flat layer of neurons. And this 3-dimensional culture was really opened by induced pluripotent stem cells because pluripotent stem cells can self-organize. Because they are close to cells that we find in the embryo, they kind of remember what it was like to be an embryo. And when we were an embryo, we were all an embryo at once. Well, we knew what to do. It's kind of sign we knew what to do. Okay? You want to do muscle, you want to do neurons, etc. And so the cells can self-organize, and if you culture them in 3 dimensions, in little balls of cells, they will grow, they will differentiate, and they will start making tissue. At the lab, we like making what we call cellular organoids. These 3-dimensional tissues that we culture are called organoids. Not because they are small organs. Be careful. Sometimes you think organoids referred to as a mini organ. They are not an organ. Okay? You cannot reproduce a mini liver, a mini brain, etc. Maybe in 100 years' time, I don't know. For the moment, they resemble an organ, which is why we call them an organoid. But it's human cells, and they recapitulate a certain number of properties of stem cell, of tissue. So we start from induced pluripotent stem cells, and then we detach them from the bottom of the dish, and we culture them in small balls of cells here. You can see these droplets here, and inside each droplet there's what we call an embryonic body. It's basically a ball of 10,000 induced pluripotent stem cells. And because these stem cells start to differentiate, they will start growing as a little ball of cells. After a few weeks of culture and a few media change, we have to add some factors in order to convince the cells to become neural cells. These very, very small balls of cells will grow. They will grow until they are 2 or 3 millimeters in diameter. Here you see 3 organoids in a dish, in a 6-1 plate, and you see that these are 2 to 3, maybe 4 millimeters in diameter. These organoids contain neurons and they contain glial cells, such as astrocytes. The brain is not just neurons and astrocytes. There's endothelial cells, there's microglia, Okay, so these organoids recapitulate in some fashion the human brain, but not complete. But still, it's an interesting model. And what's interesting is that these organoids, it takes 1 month to get them, but once you get them, you can culture them for a long time. We used to be proud because we had organoids that have been cultured for 2 years in our laboratory. There's a paper published last month where they did 6 years of culture of organoids. Okay, I think this may be the longest cell culture ever achieved. So it's interesting for people who are interested in long-term culture, aging, etc. Because when you have a model in a dish, then you can start using it to understand the nature, which is what research is about. This is what the organoids look when you cut them in slices, stain them with antibodies that are specific for certain cell types, and look at them under the fluorescent microscope. You can see, for example, in this slice that in green here there are immature neurons. You can see that these round structures here, which are called rosettes, have markers of neurogenesis. There are cells that self-regulate and grow in different states. And what we can do, and this is actually something that we've been specialized in our laboratory for a few years now, is do three-dimensional imaging. Because looking at what goes on inside the organoid is complicated. If you think of an orange, if you want to see what's inside the orange, you can cut it in slices. It takes a lot of time, it's messy, and oranges everywhere. And then you can take a picture of each slice, kind of reconstruct it. Okay, use computers to put them together. Okay, it's kind of complicated. We do three-dimensional light imaging, which means that we can make the organoids transparent and look through them and image them using a special microscope or light microscope. And using this, we have this type of pictures. Let me see if I can start it. You can reconstruct in 3 dimensions the inside of the organoid. Here you can see the neurons going all around the organoid, which was very difficult to do. Slice. Okay, this is a single organoid viewed in 3 dimensions using light sheet microscope. So this 3-dimensional culture is important first because it's closer to what you find in a tissue, in an adult tissue, in a normal tissue, and because it shows interactions between the cells that you cannot recapitulate in 2 dimensions. Okay, I'm not saying that 3 dimensions can completely replace 2 dimensions, but it adds a layer of complexity to this technology. Now, obviously, there's a lot of applications that you can think about creating a living tissue in vitro. Here are the main applications. First, you can think about cell therapy, called regenerative medicine. We can use these cells to make a tissue and then graft it to a patient. We can model diseases and understand diseases. We can take cells from patients. You can use them to do drug development, test drugs to maybe find a cure for these diseases. You could use these cells and this technology for personalized medicine. Personalized medicine means that you take a cell from a patient and then you test if a particular treatment is adapted to that particular patient. Okay? Because induced pluripotent stem cells are obtained from reprogrammed cells, I can take, as I said, the skin of one of you, generate induced pluripotent stem cells, make an organoid, and then maybe test a drug on it. And finally, these cells can be tested for toxicology testing. Okay?
because they recapitulate human tissue, we can test it for testing the toxicology of particular compounds. Now, as I said, in my lab, we're working hard on modeling diseases, and mainly Alzheimer's disease. We've been working on Alzheimer's disease for quite a while now. We have induced pluripotent stem cells from patients that have a particular form of Alzheimer's disease that appears very early during the life of the individual. And we are trying to look inside the organoid with the difficulties I told you about. It's difficult to see inside the organoid. If inside the organoid we see the same aberrant structures that we find in the brain of Alzheimer's patients. In Alzheimer's disease patients, there is a very particular form of aggregated protein. This protein is called A-beta that appears. They are called plaques. And usually these appear pretty late in the life of the individual. Now here, because you can follow the life of the organoid from the beginning to the end, you can try and understand how this aggregation appears and maybe how we can stop this aggregation to happen inside the organoid, maybe the patient model. So disease modeling is really important in the field of pluripotent stem cell research. It's true for Alzheimer's disease, it's true for a number of other diseases, some of which that we are also studying in our laboratory. But of course, there are many diseases, if not all diseases, that can be modeled once you have the human tissue in vitro. Obviously, and this is one of the applications that is the best known, maybe not the most frequent, but at least the best known, is that you can use cells derived from induced pluripotent stem cells and pluripotent stem cells derived from embryos. You can use these cells for regenerative medicine. Now, I like making this type of tables. This is a table which is more or less complete of all the clinical trials that are currently and have been done from human embryonic stem cells. You remember, they are the pluripotent stem cells taken from human embryos, or induced pluripotent stem cells, the cells that are derived from the skin of patients through reprogramming. And just 2 years back, the table on the right were basically empty. There weren't any clinical trials from induced pluripotent stem cells. And we're starting to see them, okay? These clinical trials, they're all in phase 1 or 2, so they're very early in the stage. But keep in mind, the research on pluripotent stem cells is only, I would say, 20 years old, okay? Which in science is really not a lot. So there's a lot of news coming from regenerative medicine, and you have to keep curious about them, and I'm pretty sure some of them will be pretty transformative in the next few years. Then you can think of using pluripotent stem cells in toxicology testing. Now, we've been wondering for a while if we could use our brain organoids in order to test compounds, to test the toxicity of a compound, to test the neurotoxicity of a compound. And this is actually quite complex. First, because these organoids, they're basically little spheres. I have a few organoids here. You can come and look at them after. They are little floating spheres of neural tissue. And it's great, but if you treat them with a compound, how are you going to know that the organoid is doing good or doing bad? And we come back to the fact that we can slice them, we can measure them, but there are not any techniques to be done. So we devised an algorithm that could put together all the data that you can get from these organoids and achieve a score. And by calculating this score, which compiles the size of the organoid, the number of little rosettes that we see, the stainings of different types of cells, you get a number, and from this number, you can see if the compound that you treated the organoid with is low concern, potential concern, or high concern compound. Okay. This is a publication which is currently being reviewed and has been published in bioRxiv. And it's an idea of how you can use organoids, three-dimensional models, in order to measure the toxicity of a particular molecule. Now, one main concern when using pluripotent stem cells and organoids is the variability. Each organoid is very different, and if you come and see the 5 organoids I have in this cube, you will see that the 5 organoids, even though they were all done at the same time, they don't look the same. Okay? There is variability from one organoid to the other. So you have to find ways to limit this variability. And to do that, one way is to try and control the conditions of culture of these organoids. And one way to do that is to use chip technologies. When we say chip technologies, it's microfluidic designed chips on which you can place the organoid. And you can use microfluidics to change the medium, to measure what's inside, etc. This means that you have less intervention on the organoid. The intern doesn't come on Monday morning, change the medium, and which introduces variability, and you have much more controlled variables. We've been working with a French company called UNIgreen, and there are several companies around the world working on this type of technology that devises small chips which are adapted for culturing three-dimensional tissue. Now, using all this, I asked myself, what are examples in the literature of using iPS cells in the field of environment? And I found a few papers, maybe they're not the most seminal papers. Again, environmental research is not my field, but this is what is fascinating about iPS cell research, that you can go from one field to the other. And there are a few articles that have been testing, for example, the effect of microplastics on cells derived from iPS cells. This paper tests the effect of microplastics on cardiomyocytes, and indeed they have found a decreased contraction of these cardiomyocytes, two-dimensional cells that contract in the presence of polystyrene. They've tested microplastics on intestinal epithelial cells, gut cells, where they could detect the microplastics going inside the cells. This is what you see in red in this figure taken from this paper. There has been a paper testing the particles from car exhaust on microglia. Microglia are the immune cells of the brain, very difficult to obtain. And here you can get human microglia from iPS cells. And they tested, and they saw that there was an impaired phagocytosis due to the particles from car exhaust. Now, obviously, at the moment in France, there's a lot of talk about acetamipride, which is an agonist for acetylcholine receptors. There's a petition going on because France wants to reauthorize this pesticide for agriculture. And so I looked in the literature if there had been any tests on iPS cells on acetamipride, and I found a couple. For example, one where they tested the neurodevelopmental toxicity of acetamipride on neurons obtained from iPS cells, and actually they found no toxicity in this particular assay, in this particular type of cell. And there's even a paper where they looked at the electrical activity of neurons derived from the iPS cells. And they found an excessive neural activity in the presence of acetamipride only when they were at above 100 micromolar, which is very high concentration. So no acute effect of this compound, which does not mean that it doesn't have any effect, but at least now we have the means to test it on different types of cells. You will know that these are two-dimensional tests. Okay? They are done on neurons in 2 dimensions. There are no, at least I haven't been able to find a paper testing UNIgreen on organoids. However, there are a couple of papers looking at organoids and environmental testing. Again, looking at microplastics. Authors have been looking at the effect of microplastics on kidney organoids and finding that they have inhibition in the growth of the kidney organoids, which may be reminiscent of the problem during embryological development. Again, one article does not complete a study. Okay? It has to be really repeated articles. You have to understand the effect, you have to understand the methodologies. There's also been a paper on road dust on colon organoids, gut organoids, where they found that there was toxicity of this road dust on the gut. Okay. So there's a recent review on Organoids-on-Chip for environmental toxicology, which I recommend you have a look if you want an overview of what's done in organoid environments. I think it's a field that is developing, that has a lot of potential. And even us, at the lab, we've done environmental testing. The environment we tested was a bit different. We wanted to test the effect of microgravity on the development of cellular aging. So 18 months ago, we sent 100 organoids to the International Space Station. It was a pretty complicated process because you had to ship them to Cape Canaveral, put them in the rocket, send them over there. The organoids had to be changed by an astronaut for 33 days. So it was a big study, which we are still completing the study of and will soon be published. But we wanted to test the effect of the condition of growth in the ISS and maybe the microgravity on the human brain using brain organoids. Okay, so per se, when I was preparing this talk, I said, oh, we also have been doing environmental testing, even though we have to be testing directly compounds in the environment. So I hope you will remember this from this talk. iPS cells are great. Organoids are cool. But many problems remain to be solved. First, iPS-derived cells are epigenetically embryonic. They resemble a lot embryos. Okay? And when you're like me, you're interested in Alzheimer's disease, which happens late in the life. It's kind of complicated to use an embryonic model. But I think we can understand something fundamental from the distance from this. There's also a high variability of these protocols. Every organoid is different, and you have to take that into account. And then there's the complexity of the system. The complexity is good because we are closer to what's happening in living tissue, but it's bad because we biologists, we don't like complexity. We like to know is my cell green or not green. So there's still a lot to learn, and it is in your hands now, future scientists, to use these models in order to answer complex questions. I'd like to thank the members of my lab who are working at the joint labs Biotechnology in Fontenay-aux-Roses, and all my collaborators who've been working with us on these different projects. And again, I remind you that we have a meeting in November in Lyon, where all the French community of stem cells will meet and speak in English on stem cells. So if you're interested, come to Lyon in November. Thank you very much. Are there any questions in the room? iPS cells, organoids? Yeah,
Just
I'll give you the one.
thank you. You said that you do a project or research on Alzheimer's disease. Where do you take the cells from? You do the reprogramming of them? You take them from somatic cells, or you take the system cells to
So this is the problem when you're working on Alzheimer's disease. Alzheimer's disease appears sporadically in the population. There are some genetic factors, but they're probably mixed with other factors, environmental factors probably. We wanted a straight shoot approach. So we obtained cells that came from patients that had special form of Alzheimer's disease because of familial Alzheimer's disease. And these poor patients have Alzheimer's disease starting at 25 years old, 30 years old. And we know that this is due to particular mutations in one gene called APP, and this particular mutation in its families unfortunately gives them juvenile Alzheimer's disease. So we tested that. We have also reprogrammed in our laboratory cells from Down syndrome patients. We know that Down syndrome patients is a triplication of the 21 chromosome. What's on the 21st chromosome? The APP gene, which is so important in passage. So they have an elevated number of APP proteins. And indeed, in Down syndrome patients, early in their life, you see these amyloid plaques. So we use genetic models because it's so difficult to model vitro the sporadic nature. Although we did a few years back test a molecule that when you add it to organoids, normal organoids, it's significantly increased in the 42. So there are ways to tweak the system in order to model this system. It's a complicated disease model.
do you have any have you gotten any positive results from it already?
So at the moment we are trying to measure the presence of these amyloid aggregates inside the organoids. And judging even from the literature, and there are many people with more smarter than me, and also with more money, that have been trying, and there's not a lot of studies that have had really high aggregation in vitro. There are some. Okay. Once you have these aggregates, then you want to see if it has an effect on the neurons. So that will be your next step. So I still have a lot of work on my hands. Yeah.
So there is a question on the net. It's related to the difference between your cell model and the animal model. Because of course, one of the promise of this model is maybe to replace animals in some way. And so the question is, are there any differences in the toxicity level obtained in organoids model and epidemiological studies or animal studies?
So organoids are sometimes referred to as a way to reduce the impact of animal models. Indeed, the whole scientific community is engaged in what we call the 3 R's, reduce the number of animals used in vivo modeling, refining these models, making them more precise, and third one,
epidemiological studies.
Yeah, and epidemiological studies. So we have to be careful because we are not going to replace these models by 3-dimensional, these in vivo models by 3-dimensional models. We can refine these models, we can add something to it, but I think that animal models will always have a plus because they recapitulate the physiology. Okay. So when I hear the word replace, can organoids replace animal modeling? I say, well, I don't think so. Okay? But definitely they will reduce animal models and they will replace some aspects of it. Now, concretely, organoids is a very new field. Organoids were first described in the early 2010s. So I don't think there are concrete results that have been taken that can show that they are superior to in vitro models, if that's the question. Okay. But we can certainly access some complexity that is more difficult to obtain in animal models, and some diseases cannot be modeled in animals, and these are diseases that are particularly interesting to see in organoids.
So thank you again, Frank, for this presentation about organoids, and we will invite now Torben, who succeeded in joining us. He didn't give up, and he's now with us now.
Yeah, we work in toxicology. I'm from Pepper, which is located here in Paris. I'm from Sweden originally, so I've been here 1 year now. As the chief scientific officer, and Pepper started a little more than 5 years ago because there's a gap in say trying to have methods that are working in the toxicology area of endocrine disruptors, have them approved so that industry and institutions can use them. And one of the ways to approve them is to get them into the OECD system. So Pepper was set up to bridge that gap. And we've been working now for 5 years with some methods. We take in methods, say, once per year. We take in 2 or 3 methods and start them for validation. And then the first one that has gone the whole way and sent to OECD, that was 1 year ago now. That has been reviewed and it's now being drafted as a test guideline. So that's the process that we want to cover. Okay, let's see if we have some slides to illustrate all of this. Yay!
Nice, good.
Yeah. Okay. This one?
Yeah, go on the right to see if we.
Yeah, there we go. Yeah. So if we start with the toxicology world, it's like anything can be toxic. The medicines we use, or the chemistry that we encounter, or substances, mixtures, everything. You've seen Frank testing pollutants or street things or all kind of things, but we encounter toxicology everywhere, of course, and we want to help to reduce that or keep it out of our air. I mean, if you have seen the pollution in Paris over the last 10 years, you will notice that it's gone down significantly. And that's a very good thing, of course, because in the old days when there was diesel everywhere and it smelled horrible, you knew that there was toxicity. But that has improved, of course. But we still encounter toxicology everywhere, and we need to be able to find it and document it and say let's have some limits to how much you can have, or say okay.
Yeah.
Share the right screen.
Okay. So there's 2 things, and Frank also addressed it a little bit, that when you really want to know the toxicity to us or to animals or the environment, it is not only about determining that there is toxicity, there is a risk, and we do that regularly with cell assays. We determine the hazard of something, but we also need to know what is actually the exposure, and this is where the competition between trying to go in vitro and use cells is with the animal studies, because in animal studies you can both have the hazard and the exposure, and you rarely can have that in the cell systems, and that is why it is a little bit difficult to just replace animal studies, but we can definitely reduce it when we have better models. So there are some gaps when it comes to going, say, reducing the animal use. There's toxicity has always relied on animal use and rat models or dogs or whatever to see if there's, say, toxicity involved in what you are testing. And the idea to say reduce and replace them, it has been around for a while now, but it will take a long time to get there. But they are also expensive and time consuming. And the models that Frank introduced here are quite advanced cell models and needs a lot of work and development. But they belong in a category called the NAMs, which is non-animal methods, or you can call them new approach methods. And that is a whole new concept that the research environment and the regulatory environment sort of get together and think that many of these NAMs, these methods, they can sort of help us to reduce the use of animals and maybe even be better predictor of the human situation if they are based on human models or human cells. And this is why there's a lot of efforts in this direction. So one step that I mentioned before, because I mean there's every, say, cell laboratory out there will have their own models, they will have their own research, they will be experts on their methods, but in order for others to be able to use methods, they need to be validated. It means that you must take the method, transfer it to another lab and see that that lab will get the same results. It's not always the case. It's sometimes difficult to get the same results in different labs. So in order for a method to be approved, you have to go and say, okay, let's involve 3 labs and make them do the method. They can train on some compounds that are known where we know the results, and if they get to the same results, we trust that this is going to happen, and we trust that the method can be transferred and is worth to investigate. The next step is that you send them compounds that are blinded to them. They will not know what it is, and then still get to the same results in all the labs. That is a blinded ring test. And if they can do that, then there's trust in the method, and then the OECD will say, okay, let's make a test guideline out of it. So this is the process that we are getting involved in. And those test guidelines, they will end up in the OECD homepage, and the developer lab, they will be able to assist other laboratories to set it up if they need it. There will be, usually if there's a test guideline, there will be companies that would want to have the method as a service to others. So, okay, this is an interesting method, people will use it, we will set it up in the lab, and then customers can come and use the method to test their compounds. So PEPA works in the region of endocrine disruption. This is a very peculiar area of toxicity because it's really difficult, because endocrine disruption, it means any kind of chemical that can interrupt or disrupt the hormonal systems. And those are many. There's so many different hormonal systems in us at the moment. And we, in order to, there's not never going to be 1 or 2 or 3 tests that can cover it. We need tests for all the hormonal systems to be able to detect if there's a risk of endocrine disruption. So that is a very big area that we try to cover. And we also try to cover it because there's actually very, very few methods that are approved. There are some animal studies that can go in the direction of testing chemicals in this area. And there's 2 test guidelines. That is it. This area started, I would say, back in the 80s when agriculture tried to use steroids to enhance the growth of the pigs. And those steroids ended in the water, and they changed the environment for us. It meant that some males would have low fertility. So they were detected after it had happened. But we soon realized, okay, steroids is a thing we need to control. We need to be able to figure out where it comes from and reduce that. But it's still an area that is very big. So the first, test guidelines were for estrogen-like compounds and androgen-like compounds because those were really the big things from the start. And then there's the thyroid area. If you know the thyroid hormones and how they control your life, it's worth to look into for sure. But now the area is expanding into metabolism and all other areas where hormonal systems plays a very big role. I put some of these here. They're called the EATs, the estrogen, androgen, thyroid, and steroidogenesis, because if you mix with your own steroids, then you mix with your whole body. So that's called the EATs. That was the start. And then the non-EATs pathways, they go wider into metabolism, immunology, and developmental biology, where Frank's models might come in very handy. Neurotoxicity, for instance. So we are looking at retinoids, the PPR gamma growth, some of the other steroids, and nuclear receptors. Nuclear receptors per se are very interesting because it's not that they react to one thing. I mean, they have a hormone, but they are controlling gene regulation. That means that they are actually controlling a whole program for your cells. So when you mix with them or when you interfere with these, there's a large impact. Of course, in the body, there's a lot of, say, regulatory systems that will try to help on the way and mitigate things that is going on. But it is very impactful to go for steroid or nuclear hormone receptors because they have gene programs attached to them. So it can look like this. You have a whole set of receptors. They have impact on different things in the cells, and that reads out in the tissue organ in the individual. And what is one of the supporting concepts of, say, this toxicity or toxicology is the development of what is known as AOPs. That's adverse outcome pathways. So OECD is asking researchers to point out what are the pathways you think that are detrimental. What is the outcome in the end, and what is leading up to that? So if you understand that your receptor or your molecule somewhere in this path has an impact down the line, which is detrimental, then you know what to look for. So there's a whole Wikipedia of AOPs. Now it's called the AOP Wiki. You can go and look at it. There is more than 500 pathways now described to support the toxicology and to support regulators to understand what can happen when you are manipulating any hormonal regulation. And we actually work out there in the green area where you have the receptors. We have methods that try to detect these so that we early on can say, okay, we have something that is potentially dangerous because of the pathway that it leads to toxicity. So the targets that we have been focusing on for now are in the steroid area. As I said, they could be steroid hormones. It can be the nuclear hormones. You can look at the thyroid hormones. We have methods that cover a lot of these. As I said, there's a multitude of biology associated to these. Yeah, I won't say more on this. Yeah, you can even have targets that are directly, of course, going into the biology. But if you have a target that is a gene regulation, then you have a massive impact on your biology, whereas other targets might have a more direct effect. You can also think of a very specific assay. Okay, let's go for a steroid receptor and say we make an assay that detects when something happens to this receptor. And then you know that, okay, you have something that is steroid-like. But you can also have phenotypic assays, which would be maybe some of the models of Frank, where you will see an impact. For instance, how is the growth of a neuron or the dendrites of a neuron? Is that impacted? And there can be many pathways leading to that, and there can be many toxicities inhibiting this neuronal growth, which means that it is a phenotypic assay. It is detecting a phenotype, but there can be multiple, say, pathways leading to that. So in one end, you have something which is very specific, and in the other end, you have something that is covering more biology. And both are really useful. Yeah, this is to put, say, a typical AOP pathway on top of the whole system. If you have a molecular initiation on your receptor, in the end, you won't be detecting the gene regulation. It will happen in the cell that you are using. But you will have some key events, and it leads to an adverse outcome in the end. So that is how you can think of these layers. So I mentioned in the beginning what Pepper does. We are filling a gap where in order to have more methods that are approved in the OECD system that can be utilized by the industry and to be used for submission of their dossiers, then we need to have more methods that are approved. And it's very difficult to get approval of methods because you have to go through the validation. You have to transfer it to other labs. You have to show that the labs can get to the same results. And once you've done that, then there's a good chance you can get an acceptance of it. But that's, for us, that has been at least 6 years now for the first method. There are stories of, in the old days when there was no Pepper, this could take 10 years, it could take 12 years. And that is way too slow a system when you want literally hundreds of methods that are approved so that they can be used and trusted. But at least we are doing our part. We select methods. We do that by asking people, say, do you have methods that are interesting in this type of toxicology? And they will send us the method and say, yes, why don't we try this one? And we look into their readiness, which means that is a very good procedure already described? Do they have nice results to show that it is trustworthy? Have many people worked on it already in the lab, not only one, or has it been transferred to other labs already, so that we see that there's a likelihood that it will work. We have relevance committee that actually helps to select these methods, and these people come from all over, all kind of stakeholders, the industry, researchers, NGOs, all kind of people are involved in this, so that we get the most interesting methods into validation. What we then do is that we figure out, okay, this method requires this, and then we write, say, a call for naive labs. And the naive labs that want to participate, we can pay them to be part of this. And that is helping a lot, because doing things for free is difficult. But sometimes there are labs that are interested in the method, and if we can pay them to be part of the validation project, then it's really helpful. We put say a team or a project together, which means that we sit as project managers, and then the 3 or 4 labs that are involved, they get the protocols, and they do the work wherever they are. In the beginning, it was largely in France, but now we have labs in Germany and Netherlands and Belgium and Sweden and the UK and Spain. So we are expanding throughout Europe. Yeah, so those are the areas that we have been focusing on. We wanted to expand the steroid receptor so that we cover all of them instead of the 2 that was already validated, and we cover nuclear receptors, we cover some thyroid hormones, and then neuronal development as one of the phenotypic areas. It can look like this if you say put them up. So the 2 that have crossed over there, there's GRTA and RARTA. Those are, so that is glucocorticoid receptor transactivation assay. That's the first one that was sent to OECD. And last week, we sent the first draft of a test guideline for it. So that is the one that has gone the whole way. Now it will end up with the expert groups of OECD to go through this. But this is a nice story. The RARTA, which is the retinoic acid transactivation assay, which is also a very, very important receptor. It's a nuclear receptor for retinoids, and they control a lot of biology. And we sent the validation report after a successful validation project 3-4 weeks ago. So we are going in the right direction. We have one from mineralocorticoid receptor. We have one for the progesterone receptor, and this PPR gamma, that is the peroxisome proliferation activation receptor. And there are 3 PPRs, so we are going for gamma, which was one of the first ones that was discovered, and it controls a lot of biology, particularly in the metabolic area. And it's also one of the key targets of medicines, actually. So there's chemistry that we can use from that. In thyroid area, we have 2 projects. We had functional projects, the TGA, the HGA-TGA, that is a liver cell project where we are looking at an accumulation of triglycerides, because if that happens in your body, you are on the path to steroidogenesis in your liver, which is a very bad disease to have. You can end up there if you drink a lot. You can end up there if you consume too much energy for a very long time. It's related to diabetes or obesity, for instance, but there might be chemistry that will enhance that. We can try to detect that with this kind of method. And then we have some phenotypic or omics methods over here. Neurites is one of those that relates to Frank's work. It's the neuronal outgrowth from some stem cells that we are differentiating and seeing if the chemistry will inhibit that. Our projects, they look like this. So as I said, we are we are selecting a method that we think is interesting in the area of EDs. There needs to be a standard operating procedure, or at least a well-described procedure, how to manipulate the cell, how to grow the cells, how to manipulate and how to test them in detail so that other labs can use it. And we sometimes have to help the developing lab to write that SOP in a very nice way that it becomes generic and available to other labs in a way that it's understandable. Then there's a training of the labs that join the naive. We call them naive labs because they're not naive at all, but they are naive to the method. It means that they cannot have used the method before, but they have to go to the developing lab to be trained in the method, how to grow the cells, how to manipulate, how to keep things, and how to test compounds. And then they go to their own labs, set it up, and need to get to those same results. And when they think they are ready, we go to phase 1, and then they will have like 3, 4, 5 compounds with known activities, and they should get to the same results again. And once we have done that, we can go on. The phase 2, that covers the ring trial with blinded compounds that I mentioned. So it's a much larger set of compounds. It will usually be around 30 blinded compounds that are sent to them. Of course, the safety sheets are there to be opened if something should go wrong, but otherwise they don't know what it is. They cannot know what the compound is, because if you are biased by knowing what the compound is, you're also biased for the results. So they cannot know it. They test them blind and then get to the same results. And if they can do that, that is really, really good for the method, because we trust that it's really working well. At the end of that, the labs make their reports. We do some statistics on the, say, reproducibility or the concordance between the labs, but also within the labs, because they cannot run it once. They have to run 3 times in all the modes of the assay and get to the same results. And there will be small variations. Sometimes they get the result twice and not the 3rd time, or, you know. So the whole goes into statistics to look at how often are they in concordance with themselves and how often are they in concordance with the other labs. And for GRTA, that was huge. That was almost 100%. Never seen anything like it. So that was very nice for that project. We write a validation report, and it's the validation report on the project that summarizes the whole project, all the statistics, that goes to OECD, and the OECD helps to organize a review of the whole thing. And then, of course, we update the SOP, because you learn something on the way when you do these projects. And that has to be captured so that in a procedure or the process is captured into the SOP, so that there's no mistakes or there's no misunderstandings. You can see the small euro signs. That means that the labs get paid for their work from us, because we are supported by industry and some ministries and so on. We always have a data manager on board to be able to compile all of these results, because there's a lot of results from many labs. So, and we build a database on this, and he is also paid for his work, and so is the statistician. We need to have an independent statistician, not to be biased by the results and say, okay, it looks like this. This is the conclusions, and we can bring that in. Yeah. So this is how it looks, and then it ends up in OECD, and it, as I said, it can take 5-6 years. I hope to see the first one do it in 2 next year. We have one project that is running really fast, very simple and very nice. And I think we have also learned a lot on how to organize this in a let's say a very strict way, so that we lose very little time between things, or between phases, and we'll be more, say, straight in our work. I took an example with me because that's a recent one from the retinoic acid transactivation. It forms heterodimer with another retinoid receptor called RXR, and that is a funny molecule because that actually forms heterodimers with the PPR gamma and the PPR alpha and with 15 other receptors. So this particular heterodimer has control over a lot of biology, but so does all of the other ones. So there's a lot to be discovered here. Yeah, this set of receptors, they control embryonic development, which is one of the things that early on was understood by endocrine disruption, is that you can affect the embryo when it grows. You can also affect the mother. There's cell growth differentiation impact, so that's not very good. That's very general. It has impact on bile acids, on metabolism, immune cells, and on cancer. And that is why it's actually also a cancer target. So some companies will try to develop chemistry to this to inhibit cancer development. So there's actually chemistry that can be used for the method to be tested. I think that if you know anything about vitamin A, you know that if you lack it, you can lose your sight, for instance. That has to do with ocular cells that first you grow you grow the color blindness, and then in the long run it can be. I don't think that anyone is suffering from vitamin A deficiency anymore, but it was a thing in the old days, and we know why now, because of this hormonal system. And to mention, there are actually 4 AOPs where this receptor is the molecular initiation event. So there are actually 4 described toxicity pathways already known, apart from all the biology. So that's why it's important. A transactivation assay can look like this. So you start with a low concentration, and then with increasing concentration, all of a sudden you will see that there's a takeoff of the signal. This is based on luciferase, so you measure in a luminometer, and it's very simple. And then you test something like bexarotene, and it will take off. And okay, we have a ligand for this receptor, it will take off. And then you take a normal RAR ligand, something that is very similar to retinoic acid, and you put it in there, and then you put them together, and then you see that the signal is going further. It means that not only does it trigger RAR, it also triggers RXR. So it triggers both of those in the complex, which means that it has even further implications. There's another test compound where you can see an inhibition that can also be detrimental. It doesn't necessarily have to be only that you inhibit something or activate something, it can also be the inhibitor. You are manipulating the system with this chemistry. And it's clear that these compounds have this effect. And in your normal daily life, if you are exposed to this, there might be an impact on the biology for you. And this is how we then put together the statistics of it. So each of these squares is one of the determinations for one of the compounds in one of the labs, right? So we have 30 compounds, and then you have 4 labs. They even have names here, so you can see where they are. And then we give them a green when they are agonistic, okay? And they all get to the same result over and over and again. Okay, here is something where 2 labs got one result and 2 labs got another result. That also happens. Here's one where one got a result and one got the same result, but it discovered that there was contamination. So we couldn't trust it. So usually it looks like there's good concordance between them. This is what we base our statistics on. Okay, and then to the process. We put together these tables. We present it to the OECD. We have to pay for a reviewer to coordinate, or they're not the reviewer, they are the coordinator of the review. So there has to be an independent person that coordinates the review. The review will be 5-6 persons that are experts in the area. They will be selected from a list that the OECD put together. And then they go away, read our report, look at the things, they come back with questions to us, and then they deliver a report with a lot of recommendations and criticism and this, that, and the other. But we have to say, look at that and take that into account. We make a reply to that. We update the validation report, we update the SOP. And then it all go back to OECD again. So that now it will end up in the OECD process and hopefully can become a test guideline in some case. That's the idea. Have I covered this? Yeah. Yeah, exactly. So if there's positive feedback on the report, then we can continue to the test guideline. And this is all because we need these methods for going into the world of toxicology and use them. And that's the next step after this, because when, say, medical companies or chemical companies, they want to have their chemistry approved, their cosmetics, whatever, they need to be able to show that there is no toxicities. So they will use the method, test them and say, okay, it looks nice, there's no toxicities. I will put this in the dossier with hundreds of other results. And then they send it to the regulators and say, we have done all of these tests and it looks nice. There's no worries. Please approve my chemistry. And that is the process that follows all of this. So you have substances, you go for a battery of tests that you test your chemistry in, which is relevant to what you're doing. If it's different from agriculture to cosmetics, let me be sure that, and medicines, that's really another world. Then all of the data together, they might be used in what is known as weight of evidence. It means that if you pull together a lot of data from a lot of methods and they show similar things, then you have good weight of your evidence that it's fine. Of course, as Frank also said, we are not getting rid of animal studies. There will be data from animal studies also. But of course, the more NAMs we can use, the less we need the animals. So that is the whole idea. For cosmetics, they cannot use animals anymore. So don't believe that they do that anymore. It's 10 years ago that animals were abandoned for cosmetics. They have to use other methods. And they will use a weight of evidence. They will do a tiered approach for their chemistry to be approved, but they're not using animals anymore. And then it goes through a submission and then an assessment by the regulators in whatever area that is. So that is the context of what we are doing in one end and how it is being used in the other end. And then, so of course, in order for this to happen, the regulators also need to accept that the results from these methods are good and valuable. So there's a new thing, because they are used to the animal studies and used to the regulation around that. and they're not so used to having all of these new methods coming in. And so they need to be educated and understand the impact, and understand that, for instance, if you have very advanced models like Frank's models, they are probably more predictive of the human situation than a rat model. If you have stem cells or thyroids that look like the human situation, they are probably better models than a rat model for many things. And then you have to add some exposure or say in silico modeling on top of it. We need NAMs that are validated. We need to understand the relevance to humans, or if it's in the environment, the fish or the birds or whatever. And we need to have some in vitro exposure data, ADME data, CREVI data. And we have to provide a mode of action. We have to say, this is the way that this works. And if there's a relationship to the AOPs, then there's better understanding of that mode of action. So this is where the work is going on now. And we are part of a very large EU project called NAMWISE, which is trying to map how is the methods NAMs, how are they developing, and what is needed for them to be taking off and be used much more. That is the purpose of that. This is some of the people, the sponsors we have. So I have to thank these. Apart from the 5-6 people that we are, we are not very many. But these are the current support we have. Thank you for your attention.
Thank you very much, Torben, for this presentation. So we have plenty of time for questions. Are there some questions in the audience?
So PEPPER is an intermediary organization which is destined to propose tests to the OECD. I don't know if people here knew that was possible, and you really convinced us that it was important in order to reduce the time if we want to safely move. I think the work of that is really important. Is this a process that has always been there? Are there always some intermediary organizations that propose tests to OECD, or is this a new venture?
No, this is a new version, I would say. There's always been the possibility to want to have your test become a test guideline with OECD. And sometimes you can organize it yourself, you can pay it yourself. I know of companies that are really interested in that. They paid themselves the expenses of the other labs. They have included a manager and a statistician to do the work. They have organized all of this themselves, and it's taken much longer, of course. And it's very, very few that will do it. Most companies will look at this process and say, no, that's too difficult. It's too time consuming. It is insecure, and I don't know if I will get to a good result. So there's a lot of risk taking. And if you invest in validation and it fails because the concordance was not good, you have wasted maybe 5 years and €1 million. So many have been reluctant to do this at all. Academic groups are very reluctant because they live from, say, doing research and publishing in papers, and spending time on, say, many years on validating assays is not something that they are often willing to do. So there has been this barrier, saying going from a good method all the way to have it validated. That has been super difficult, and there has not been. I mean, you can go to Ekvam and get help, but you cannot get them to sponsor it. And that's the difference. Now there is a process where we take care of the whole organization and we can sponsor the labs that are involved. That is new. So there's never been something like this before.
I have a question concerning synergistic effects or effects. So you mentioned, I think, in one of your results that you combined two molecules that were serving the receptor. So this is already an approach where you try to see two molecules together.
Yeah.
But obviously, in the environment, you have huge cartels.
Yeah.
With some kind of traces that are not active per se, but when they are combined with ingredients, then we get different. How can we address these problems? When there's some issue that we can look to target some consequences.
That is very complicated, of course. What you can do is that many of these methods can be used with samples. So if you go and take a water sample from a lake and say, okay, let's see which steroids are there, then you apply 5 different steroid assays, and then you can see what happens there. So you will see the combined effect of your sample, but you will not be able to say it's because of this, this, and this. So that is, but you can detect it. You can detect that there's an activity in the combination, right? But you will not understand exactly where it comes from. That is something to be detected afterwards. But many of the methods, if they are robust enough, they can handle, say, diluted earth samples or water samples or mixtures, or for instance. You mentioned one of these from the agriculture that you were looking at. I've been involved in a project that with King's College in my previous life, where they looked at glyphosate. Glyphosate is also controversial. It's been used by the agriculture for a long time. Some claims that it is genotoxic. And if you take glyphosate and test for genotoxicity, it doesn't show up. There's nothing happens. But sometimes if you take the formulations, because glyphosate is not put out on the field in the agriculture as glyphosate, it's put into a pill or in something else. And these other formulations, they are toxic to the cells. We can see that. So, and there's differences between the different formulations. So all of a sudden, maybe it's the formulation itself, but it could also be because glyphosate is together with something else. I don't know. Cannot answer exactly what it is, but you can see that the sample that is in a combination has a toxicity. So you have to relate to that. The problem is that if you're an NGO that wants to get rid of glyphosate and you go out and say it's genotoxic, and then the company says, yeah, but we have 200 papers saying it's not genotoxic. And they are right. It's not genotoxic on its own. But some of these formulations are toxic, and we need to dig into that. So the claim from the NGO becomes wrong, because per se they're not right what they claim, but they might be right that the formulation are still not good. But they need to cover that in the correct way.
So just to be clear about it, because I wasn't aware about this. So when a company wants to bring a pesticide on the market, they just have to show the result with the active compound, not the formula.
That's probably how it happened, yeah, from the start, because glyphosate is something that has a specific activity, and when it rains, the water will dissolve it and it will go away. But some of these formulations that it's put into, they are secret, and they could be the problem. But this has only just started this investigation. But I get all of these NGO mails that glyphosate is genotoxic, and maybe not, because that's not what's been shown. But it's not the end of the story, right? And that can go for yours as well, what you are investigating. You might not see anything from that, but when it's used, it could be something. You have to have the whole context.
Yeah, and I think having the whole context is the key component, and there's a lot of debates.
Yeah.
And I think this idea of synergistic effects is really essential, and unfortunately, it's really complex.
Absolutely.
Because you can test, as you said, glyphosate and see it's not genotoxic. But as you say, is it not even know the story? Because if it mixed with something good, genotoxic or toxic environment or something, it's really difficult to see. You cannot test everything. And I feel like because everyone is rightly so scared for his own health and the health of the environment, but we cannot test everything. We live in a world of chemicals, whether they are natural or not. And so I find it really difficult to see what we should test. And do you feel like the REACH legislation, which necessitates every company to test the compounds it sells in itself, is it one way to answer these questions?
In the more results you can get from tests of anything, the better it is, of course. And you need the methods to do that. But I mean, I agree that it is more complex than just taking one molecule and test it. In medicine, that is what you do. You this medicine, we need to get it out because it will cure whatever. And you have hundreds of pages of toxicology before you go into clinical trials, and the clinical trials and follow the toxicology just as much as the efficacy. Right? Sometimes it's all about the contents of it. Okay, I take this medicine, but I also take this medicine because now I need that, and I take this medicine. So I take 3 or 4 medicines when I'm 80 for different things. And the combination of these, you need to understand. You cannot understand it for chocolate style to cellular. The same thing is for the environment. Once you have a load of things together, there's toxicity to be exposed.
So what we're saying is that this is in pharmacological assays, there's a phase 4. Once the molecule is out there, then the doctors come back and say, oh, I've noticed this or that.
Yeah.
And what is missing here is phase 4, but
for the environment. Yeah, absolutely. And we see it only once the toxicity has happened. Yeah. It's too late. So, yeah. Okay. We can debate a lot.
It looks like a race. You are first the pollution, and then you try to run after the pollution to find the right test to show that there is a problem.
Yeah.
We have a question from the next. So Anne Le Goff, who is asking 2 questions. So does the battery of tests change for each EDC, or is it a common battery?
No, so you would not have one battery. You would probably be able to select different relevant methods. If you are developing steroids, you would say go for the 5 steroid receptors, but you might go for a couple of phenotypic assays where you think it's relevant, and then say, okay, let's combine of these, show that there's no risk or little risk or no. But it will not be the same because in EDs, the biology is so diverse that it will be impossible to cover, and that makes it a little difficult, of course. And that is why we need all the more methods. The more methods we can get into the system that can be used, the better it is, and we are way behind. So yeah.
Are there any endpoints in germ cells or only in somatic cells? Have more precision on this question?
Depends on if it's for Frank. I don't know.
Yeah.
I mean, we have no models that are super advanced like his. So we have cell lines for most of what we do, but we also have a few models where we have progenitor cells that are differentiating, which means that they are going from a stem cell situation into becoming a specialized tissue or specialized cell. We are using some cells that differentiate to neurons, but they will not only be neurons, there will be 30% neurons, and then there will be the other supporting cells in the system. So we have a little bit of both, but yeah, for most of the part, it's very straight. Yeah.
Maybe a last question, or if you have more, can proceed. So you mentioned that we have to develop as many methods as we can. So for me, it sounds like we have to run faster. What are the engines? Where are the leverage? Who has to push?
I mean, there's a lot of push. If you Google NAMs and the world of NAMs, there's been 10 research consortia already supported by the EU or other agencies. There is a lot of different, say, cross-country, including NAMWise that we're a part of, but they are mapping out the landscape, the needs, the lags, the holes, all of what is there, and case studies, and we want to have material to use in education, both as for students, but also for regulators and others. So there's a lot going on, but to get to the end, it will take many years to get there. And it's not only about getting a lot of methods approved, of course. It's also to get them used in the right way. And that is it's a funny thing because of the rules of the cosmetic industry that they cannot use animal studies. Their approaches will be mimicked by the other industries now, because they were the first that were forced to use only in vitro methods. And they know, well, we need all of these methods. And one of the first methods that really was a big step was a skin sensitization assay, that is an in vitro assay that was developed and approved by OECD and used a lot. So, because you put it on your skin, of course. So if there's a method that can detect that this is going to be difficult for your skin to tolerate, then this model will predict that. And that is one of the success stories of it. But there's a long way to cover everything, of course. But the cosmetics industry's tiered approach will spill into our other industries, I think.
Thank you. It's a very good illustration of all the complexity of the problem we are generating in our societies and the amount of effort we have to bring. So, is there any other question, intervention? So we can thank again our speakers today. We appreciate a lot the discussion and the presentation. Thank you again. So to close the session, I just have a QR code to propose to you for survey, because we try to improve every time. So just share it on the screen, and thank you again, and hope to see you in the next webinar coming in September.